exciting moment right now.
Science Month finale.
There's a cap off to the Science Month thing. So we have 3 scientists on the line. We will get everybody to introduce themselves. What we'll do is we'll start with Ross, then we'll go Richard, and then Sttephanie. Okay, Ross.
So hello, I'm Ross Gillanders. I'm a senior researcher at the University of St Andrews in the School of Physics and Astronomy. My background is in optical sensor development for specifically, in the past more for water quality and food packaging as well. The past 7 years or so, I've been developing optical sensors for explosives, for landmines, and for IEDs as well. So a large part of what we're doing at the moment is actually using honeybees as well to collect explosives around suspected landmine areas. In addition to that, I'm trying to spin out a company at the moment for water quality sensors.
I guess it's me. Hi, I'm Dr. Richard White III. I am a computational molecular synthetic virologist. I'm an assistant professor at the University of North Carolina at Charlotte. As mentioned before, these are my personal views, and a little small disclaimer: they do not necessarily represent the University of North Carolina at Charlotte or Raw Molecular Systems Inc. This is my own personal thoughts, and we're having a conversation in the context of freely communicating about various parts of science. Mm-hmm. My lab specializes in all things viruses, from pandemic viruses to viruses that alter biogeochemical cycles in something like water or soil, or even viruses at the origin of life and how they may have triggered life and the generation of life as we know it on this planet. It may be elsewhere in the universe. So that's me in a nutshell.
Awesome. And yourself, Sttephanie?
Hello. I'm Sttephanie Shuttler, and most recently I was at the North Carolina Museum of Natural Sciences, but since then I have transitioned into being an entrepreneur, actually, and really focusing on science communication. But my research, I've been in research for close to 2 decades, and I would say my main areas of research were animal behavior, and specifically on African forest elephants, understanding their social structure. And then at the museum, I worked a lot with a camera trap, so understanding specific species of mammal, but also community dynamics. We had projects all over the world.
Awesome. Now, just to explain to listeners that are checking in, this is a bit of an experiment for us, an experiment for the 3 scientists on the line as well. So we're just looking to see where this goes, and that should be awesome. Usually our conversations consist of us asking a lot of silly questions, and although valid, we could have...
We think they're valid.
I think we could have smarter questions asked.
Yeah. And it'll be interesting to see where people in different areas of science go with the questions that they ask. So I guess we'll throw it over to you. Does anyone have any questions for anyone else?
I'm really curious about the honeybees. Like, what do you have them do? You have them detect landmines?
Yeah. So what we do is, what I work on is, as I mentioned, it's optical sensing. So it's optical chemical sensing. So we have these layers of polymers or polymer composites with other things. And if you hit them with light, say blue light, they give out green light. And if it comes into contact with an explosive like TNT, the light level drops down, and that's what we're trying to detect, basically. So we got funding 8 years ago or something like this, it was a big EU project for landmine detection. And I built a prototype landmine detection system that was about 2 kilos or something. And the problem is, though, that when you have deminers going out, so I mean, obviously everyone knows what landmines are and how dangerous they are. They're non-discriminatory and they last for a long, long time. I mean, you've got mines that are buried for 20, 25 years that are still going off, no problem. Deminers at the moment, they go out very, very slowly with dogs, with metal detectors and all of these kind of things, but you're still going onto the site, You still have to stand where the landmines are and very slowly go up and down, and it takes a long time. And It's very dangerous, very expensive, and all these things. The problem with dogs as well is they're very, very good, obviously, but dogs think they're playing a game, and you can only use them for 15 minutes at a time, and the upkeep for them is very complex as well. So anyway, so the idea was thinking, how can we, without any human going onto the site to try and find a landmine, how can we pick this information up? So with some collaborators of ours in Croatia who are bee experts, there's Professor Nikola Kezic, who I think 30 years ago at least, he'd looked into doing this with Chernobyl, so the radioactive fallout around Chernobyl. Obviously it was quite large area, to see if they could let bees go in, and if they come back collecting radionuclides on their bodies, you could analyse that and say, well, this area is still contaminated. So it's the same concept as that, really. So what we do is in this project we have at the moment, which is NATO funded, we just let the bees fly around. And they're really hairy, the bees, so as they fly around, they electrostatically pick up TNT and DNT and various other things. It sticks to their bodies, it comes back into the hive. And so inside the hive entrance, we put these little cartridges with what's known as a pre-concentrator on it. So as the bee walks through the cartridge, it deposits the explosives it's picked up, and then we analyse that with our sensors. So the idea is that if you have an area that's, say, 5 square kilometres, instead of having multiple people going on that site and walking up and down for months and months, you can send the bees out to just fly. They're obviously too light to set them off. They come back in, and we can say that in that area there's definitely landmines, and you can gradually, decrease that. So, but in the other side of the project we have is you can train the bees. So the idea of our project is that we say there's definitely explosives there, and then the other partners who are in Bosnia and Herzegovina as well, so they train the bees by putting them inside a tent. And if you get like a sugar, say if you get a dish with TNT in it and put a little jar of sugar water, and you keep the bees inside a tent, as they fly to get the sugar, they're associating that reward, like a Pavlovian dog kind of thing, they're associating the smell of TNT with getting a reward. So you train them for a few days, and you put them out on the site that we've determined has explosives on it. And then you just let them out, and you follow them with a drone that's got 2 cameras on them. One's a high-def camera, so each bee has to be like 10 pixels at least, and IR as well. And there's some really fancy image processing I couldn't even begin to explain how it works, but they can discriminate between, say, a bee and a fly, or a bee and some grass that's swaying and that kind of thing. They strip all of that out, so then what you've got is you can just see tons of bees flying around and congregating around where the landmine is. Then you can go in and get rid of it in whichever way, So it's just a really cool project, And the problem has been that obviously with the pandemic and things, we've not been able to trial, because that's a very field work heavy project for us, We've not been able to go out. Obviously, the last time I was out in Croatia where we do the field trials was September 2019, so we've just not been able to do any proper field work. Some of our partners can go out and do bits and pieces, but to do the full, field trial itself, it's been really difficult. So it's a pity for that, because the project finishes this year, but hopefully it's going to make a big impact on landmine detection, because it does work. It needs a lot more validation, because and maybe Richard as well would know this, if you've got a spin out or a startup going, but if you're validating instrumentation or any kind of methodology, it's really, for medical devices, for example, it can take a long time. It's the same for this, because for landmines, if you're telling a farming community, for example, this area is all completely free, go and start to use it, you have to be 100% sure that it's,
right.
that it's landmine free, But the bees are really good. We have had quite a lot of press attention with this, which was unusual for me, because when I worked in water quality stuff, water quality instrumentation, you tell somebody and they just kind of, okay, whatever. But people are interested when we talk about landmines and these kind of things. But again, you get a lot of skepticism for this. And you mentioned that you're a science communicator, you're involved a lot in science communication and things like that. And which I think is really important because of this, because people can be really, really skeptical about what we do, for example, and I understand it does sound a bit wacky to use honeybees and to find bombs or whatever, but it does work, I do a fair amount of outreach and stuff as well, and it's I think it's important. Even you see now things like I don't know, about vaccinations and stuff, and people are quite resistant to it, but that's why I think people have to be, do a lot of science communication and outreach and stuff to try and get the public to understand that scientists aren't a big homogenous group who agree on everything, and it's quite the opposite, as you know.
Have you heard of the African pouch rats that sniff out landmines too?
Yeah. Yeah, it's pretty good.
So are they better than bees or worse than bees? How do they compare?
I'm not sure. I mean, the thing is with the sniffer animals like dogs or the rats, and I've heard of elephants doing it as well, but I'm not sure how feasible that is. But they are animals that are effectively playing a game because they're expecting to get a reward at the end of it, so they can be quite temperamental. I'm sure that dogs, if they don't want to play or they don't want to go out, they don't want to,
I can't train my dogs.
Yeah. Yeah, they can be quite, we always had spaniels growing up, so, they can be a bit, what's the word, individual, shall we say, but, so I mean, I think there's scope for all of them, because, I mean, we're not saying that we will replace dogs or anything, because the thing with bees are, I mean, we just published a paper 2 months ago or something where we kind of estimated how much explosive can a single bee collect, and we calculate about 35 nanograms, right? But if you're talking about, a hive that has 15,000 foragers that are going out 10 times a day each, that builds up really, really quickly into milligrams per colony, which is all great, and the bees are drones, they don't, they're not, I mean, the bees just go in and out, they're not temperamental, The only thing that's a problem is the weather, really, so they don't like the cold or the wind, and things, and obviously then during the winter, they're basically not active, so it is more of a summer thing, whereas dogs, I think, can work all year, and the rats as well, presumably. How did you move into the science communication?
Because I was always driven to work on wildlife because I loved conservation and I wanted to have an impact and, like, as many kids say, like, save the world and save species. And just the deeper and deeper I got into research, I just realized, and this sounds kind of stupid, but that, like, I mean, really, like 98% of the problems are human caused, and the solutions are really human behaviors. So with the animals I was studying, forest elephants, their biggest issue by far is being poached. And if you leave elephants alone, especially the forest elephants, they actually have a lot of habitat, even though like you fly over and you see like all this canopy cover of trees, when you actually look at like what's going on there, there's all these different oil concessions and logging concessions, but they can live with those industries. But it's the poaching that's really killing them. And so that either really involves educational messages, or if it involves science, it involves social science. So I just became really interested in, and I did dabble in some social science stuff when I was doing my postdoc with camera traps, and I worked with kids. Actually, we did a citizen science project. So our project was to incorporate our research into classrooms to get kids to collect the data. And I started some social science work, and I just really realized that it's so hard to change adult behaviors and attitudes, but if you get them while they're young, they're much more open to it. And just so much of these pro-environmental attitudes and behaviors are instilled when you're children, and they're driven by these connections to nature. Yeah. So yeah, I just started to get more interest in that, and I really like public speaking. I really like writing. So it just felt like a natural shift towards me, for me.
Cool.
So if I was going to pick on a central theme that we've been talking about here between the three of us, the kind of a central question and a central theme is in the environment and our relationship with that environment. And so being a virologist, as we encroach further and further onto new areas where humans have not been, or we're going to natural habitats where we shouldn't be, the spillover of a zoonotic disease causing a global pandemic is very, very high. And we've seen that recently with SARS-CoV-2, the coronavirus pandemic. We've seen this with likely pandemic influenza. 1918 influenza was originally all influenza viruses come from birds. And it results as well from the way we deal and handle the environment and the way we deal and process things like agriculture and land management and all these kind of things. So as we, three very diverse scientists, here, one is a wildlife expert, one is a physicist, and then the other one is a old school virologist. So how do we, but you've hit on some very good central themes here, Sttephanie, and that is, it's a people problem.
Yes.
Right. And so, how do we go and convince people that we need to change our current interaction with nature in order to preserve not only ourselves but life on the planet as we know it? And we see that obviously we're dealing with a pandemic right now. I'm working against the next pandemic, which is multi-drug resistant antibiotic resistance and antifungal resistance. But then there's this kind of looming Goliath, if you will, that's in the background that we've talked about for 30, 40 years. And that's climate change. And it's really not even climate change. It's climate disruption is the better word to describe it. And that, again, is fundamentally related to how humans deal with our environment. So what are your thoughts on what we can do better?
Me?
Yeah, just whoever wants to talk first.
Everyone.
Yeah.
Do you mean in messaging?
I'm just saying you mentioned this also fundamentally. You mentioned on some two very critical themes.
Okay, I will.
Said. One of the themes is that we have to educate better.
Yeah.
The other theme is that it's a person problem, and then how do we change? Because if it is a person's problem, if it's our problem, then we have to convince the minds of not only young people, but also people that are in a seats of power, and people that are adults. And you said it is harder to convince adults, and
yeah,
but I think we have to do something relatively rapidly, because if we don't, in my kind of general thought, I mean, most species don't make it. 99.99% of all species we've ever seen, they just don't make it. And Homo sapiens might be the species that takes it all with them.
Yeah. Right? So, I think, of course, it depends on the issue. But I think a lot of scientists just say education. And I think, just blatant education, here are the facts, that has failed us. So I think what we need to do is meet people where they are and embed the types of messages or stories from the data. So yeah, we look at data and we do experiments, but frame it in a way that aligns with their worldview. I think that's largely where scientists have failed, especially with climate change. So, here in North Carolina, farmers know that climate change is happening, whether it's human caused or not. Maybe they don't agree with that, but again, I think it's aligning it with issues that people care about. Even with talking about climate change now with the past election, I still think that people think that climate change is all about polar bears, and which I love polar bears and I care about them. But if you're struggling, living paycheck to paycheck, or you got you got let go because of the pandemic, you don't care about polar bears. You care about jobs. So I think a lot of it is about framing it in terms of what people care about. I think the other problem is I think scientists have to look at ourselves as a delivery system. I think that I don't know if it's on purpose or not, but I think sometimes we just have this air like, I know because I'm a scientist, and kind of like this better than you attitude, or at least that's how people perceive you. So part of my science communication, and I know a lot of my friends who are scientists too, is really like humanizing us. So, showing us doing just like regular activities or even just like what we're wearing, because like I said, people have this like idea that you're this extremely serious person in a lab, all this stuff, you're really intimidating. And we just need to be more relatable, I guess.
Yeah. I think that's a really good point. Again, when did, and I guess this is a question I'll ask everybody, when did scientists become the enemy? Like, when, in the time of, when did, what was the switch from, Dr. Salk, amazing, here's this new vaccine, I'll take it right away, to, oh my God, vaccines have trackers and all this stuff, and it's going to, do it. When did it change? I mean, Dolly Parton, for Dolly Parton, beloved Dolly Parton, put her own money into that biotech company in order to basically fix that vaccine. And
she turned down a statue as well, didn't she? Yeah,
and so here's the thing. We basically landed on Mars twice perfectly with these two vaccines in the last year.
Yeah. No, the vaccine is a huge accomplishment.
And yet it's seen as something, one more thing. It is seen as not what it should be. And so what happened? Somebody tell me this.
So I'll talk about the two main issues we've been talking about, which is climate change and vaccines. For climate change, you're definitely fighting against money. So I know there have been money and messaging into one party over another one to favor industries that would essentially go away as we move towards a greener environment. So there's a lot of, like, when I mean good messaging, a lot of messaging that is effective, and maybe it's sending the wrong message, but they're effective in changing people's minds or getting in the right places too. Like churches, most scientists I know, if not all of them, are atheists. They don't go to church, so they're not associating with these people. And we have disagreements about how life evolved and everything, but most people who are religious don't not believe in evolution. They just don't believe in the human evolution part. Like, they believe that species are evolving now, or like virus, or like bacteria are evolving and stuff. I think it's hard with that money issue, and I think that's part of where it's gone wrong. And that, again, scientists, if they really want to work on an issue, or conservationists, I should say, they're going to have to do things like go into the church and not present charts, but pull Bible verses and talk about, God instructed us. I don't know.
I don't know.
God instructed us to take care of the planet. I know there's verses like that.
But at the end of the day, so that's a hard divide. Now, I agree with that. But people that can separate their beliefs in Christianity and still trust science.
Oh, yeah. I'm not saying you can't do that. I'm just saying that most, at least from my experience, most scientists are atheists, and therefore they're not talking to people in these communities. So people who are religious are getting other messages, and there's no scientist in the congregation to be like, well, actually, and let's look at it this way. With vaccines, I actually think vaccines are super interesting, because I think a big issue that's going on that contributes to this not trust of scientists is that there's so many obscure health problems going on now. And I know this because I suffered with this. I had like massive fatigue problems. I had migraine headaches like crazy. And I found out it was due to mercury. I had my teeth fillings were eroding, and I had mercury poisoning. So I think that there's like all these chronic problems or rise of problems that are hard to diagnose, hard to treat. And I think people are just getting frustrated with doctors that I don't think, at least based on my experiences, doctors here in the U.S., they don't listen to you. A lot of times you're rushed. As women, and I know people of color, there's studies to show that doctors don't believe you when you say certain symptoms. So I think it's kind of a backlash to that. Like, people are just looking for, like, what is the solution? Why is this autism happening to my child? No one can explain it, essentially.
If we don't get a handle on this, obviously, we could go, if we get into more of and I don't think it's an anti-science movement. I think there is a pseudoscience movement in the United States that has always been there. I mean, at a time.
Well, it's not just the States. The pseudoscience things everywhere, man. Yeah.
So even Isaac Asimov commented on this. It's like they called it the cult of idiocracy, the cult of ignorance, right? The cult of ignorance is what he called it. And so, again, it's education. And it's also, we have these new powerful social media devices that allows us almost all human knowledge. And it also allows you to broadcast to basically everybody. So Europe, I think Finland has done a really good job. Is they're starting to educate kids about how to actually spot pseudoscience early on. So it goes back to your original thought, Sttephanie, which is we have to get it into kids earlier, but then how do we help the adults as well? So it's just a question I have. How do we make science, how do we get away from scientists not being the enemy or some deep state conspiracy or some other nonsense to being the buddy, your best buddy, you know?
It's interesting, hearing.
I want to be your buddy. I don't want to be your enemy.
Yeah. I mean, it's interesting to me hearing it from the American perspective, because, for example, like Scotland, or in general, is not very religious. We're a fairly secular, atheistic. I don't think I know anybody that goes to church on a regular basis at all, and probably the two other lads don't either. But we have the same problems with this, in terms of pseudoscience and things. And I think, Richard, maybe if what you'd said about things like social media. I mean, we all know the problems with Facebook, for example, and a lot of, there's just an absolute explosion of stuff where something that's been published in Nature is on the same pedestal as something that's been published on, naturedietnews.net,
So I think you also hit a really good point here, and I've talked about this before, preprinting. Preprinting is this idea that before it's published, you put it out there for the people to consume. And I think the news in the era of COVID-19 is basically saying that these things are like regular peer-reviewed paper, and they're not, and so they can get a lot of things wrong. Yeah. A lot of the stuff like hydroxychloroquine initially was like, okay, well, there's this really awful preprint that comes out, and it's not shown, and so everybody starts buying hydroxychloroquine, right? I mean, we didn't have this during the HIV epidemic. We didn't have preprinting, we didn't have social media. And so, but that was a problem even in that time, that, papers that were kind of on the line, new drug for people that are suffering, people will want to, they used to have like buyers clubs and stuff like this, right?
That's right, yeah.
I think a major problem is how science is taught in school. To be honest, I didn't know how science worked until I started grad school, even though I did, I volunteered in research labs. I just didn't understand the whole process, and I actually did a whole podcast episode talking about like peer-reviewed journals. So even just like the whole like asking questions and collecting data and data analysis, like you're taught this simple concept, and then in school when you have labs, if you do have to come up with your own hypothesis or question, it's about something that's like already established for you. And they want you to like get at this one thing and to ask it correctly. And then you do the experiment. And for me, the experiment would usually fail, because like I'm a kid and you mess up or something. And usually it's to demonstrate a concept that you're learning in lecture. So for me, I just really felt like science was all about like understanding facts and memorizing facts. Even to this day, like people will say, oh, you're a scientist, like you should know this. And it's like, no, like I know how to do research. I don't memorize like all these different things, or I'm not, I don't have this like wide knowledge about all these different species in the world. So I think people just don't even understand the process of science, and of course sample size, and then of course talking about the different journals. I mean, even the journals can be hard for us to navigate. That I know. I mean, I know in my field like what are top-notch journals, and then also like funding has changed science completely as well. So like some things that are just like really basic can't get funding because it's not it's not theoretical enough, or it won't be novel enough. So
yeah, I think there's from in my experience anyway, there's a kind of a sweet spot, because I'm an applied physicist, you know, and a lot of what I'm doing is, you know, building things or putting things out in the fields rather than just looking at something that works at 0.1 millikelvin, you know, in a vacuum and all this stuff, you know, stuff like that. And I struggle to find funding as well, because it's too close to. I don't know if either of you use TRL in your work, technology readiness level, but something like TRL1 would be something that's just a theory, an idea. And as you build it up, it goes up to TRL7, which means it's ready to go out into the world, whether that's a commercial thing or whatever it is, you know. So with the funding that I target, it has to be between kind of 3 and 5, you know, so something that you know definitely is going to work and can definitely be, you know, taken forward with this funding, you know, but not something that's going to be on the shelf 2 months after the grant finishes, you know, that kind of thing. But yeah, I mean, things like journal papers and stuff, yeah, it is hard for us to navigate, because even the things like the explosives detection with bees, that's taken in biology and optical physics and material science, and you know, and it's difficult to find a journal that will accept that, even though the studies are quite robust, you know. So we tend to go for environmental journals, because they do tend to be a lot more multidisciplinary, you know. But yeah, so stuff like that. Anyway, it's and as you say, it's hard for us to kind of navigate that and to explain it to each other, you know. How can we explain it to others? But I think like people, because earlier on when you were both talking about things like oil industry, you know, or having, you know, this big money that's behind this. I was thinking about, you know, tobacco, for example, where it wasn't until I think it was the 70s. I mean, everybody smoked, you know, and in the 70s or 80s, you know, so I wasn't around, you know, but somebody who'd went, no, this is definitely not good for you, you know, and a lot of people believed it, and now obviously smoking is a lot less prevalent than it was 40 years ago. So people bought it then, and I wonder, was it a case of, well, for full disclosure, I smoke, and is it a case where, you know, you're smoking and you start coughing and stuff, and you think, yeah, maybe these guys do have a point, you know? So is the analogy or is it analogous that if people now say in Austin, in Texas, are starting to see, you know, 2 meters of snow, that's now starting to personally affect them. So maybe they will say, or I don't know, what do you both think? Do you think people will say, now that I have been affected by my house being snowed under in Austin, Texas, maybe climate change is something that we really have to address pronto, you know?
I mean, I do think that the U.S. is finally changing, and polls show that there's more people in favor of taking climate action. And this last election, it was really the first election that I've heard climate change talked about pretty frequently. So I do think that it's changed, and I mean, I have no evidence, but I would imagine just the severity of the storms and how frequent the storms are, even for people who don't experience it. Like when I'm not super old, but when I was growing up, I remember when there was a big storm, it would be like once every 5 years, 7 years. And now it's like we get multiple storms every year. I just wanted to say one more quick thing about science is that people can't read graphs either. When I worked at the museum, I was advised by the curator who ran our talks and public interaction not to just slap up a graph on there, and actually not to use graphs. And if I did use a graph, I had to like slowly walk them through it. Like here's the y-axis, and this is what is going to be here, and like, you know, revealing it like in stages. So yeah, we've lost that critical analysis, and it's just all about memorization now. when I taught non-majors for biology, I was furious at the class. I was a teaching assistant for it, so I had no control over the labs. But here I was in my PhD, and I actually used genetics for my research. I used Hardy-Weinberg equilibrium to determine if we used certain markers, but we ran it through a program. We didn't sit there and calculate it by hand. And we were teaching students how to solve for P and Q. And I'm like, they're never going to use this ever again. The second they are done with the exam, they're going to forget all this stuff. And we should be teaching them like give them two different journal articles. This one says this and this one says opposite. How can you evaluate these two different studies? Things like that.
I do a lot of things with not to cut you off, Ross.
No, sorry.
So I do a lot of things with I have this thing now with my group called Ugly Figure of the Week, where we go and have them
I like that.
and the ugly figure and pretty figure of the week. And we can't make fun of, they cannot make fun of the authors. They can't say anything about the authors. They only throw the figure up there, and we have a good figure and we have a bad figure. And then why do you like this one? Why do you hate this one? And some people will say, well, I don't think it's that ugly. And then we have a discussion about it. But what can you obtain from this information? What can you get from this figure? What could you do to make it better? To make it to where it's clearer.
Do you use that for graphical abstracts as well?
I haven't done it for graphical abstracts. We literally just go out in the literature and find beautiful figures and terrible figures, and we break them down. And one of the big there's a couple that are big. They show up a lot. So Venn diagrams are a one that shows up a lot as a figure that there's too much information when people usually do more complicated Venn diagrams. They're prime suspect of awful figure, right? But they can be done well. I'm not saying that all Venn diagrams can not be done well. They can be done well.
And they have two circles.
You'll see some that have like 20 different things, and we saw one.
Sorry, can I
day,
quickly butt in? The two non-scientists here are a bit confused about what a figure is. What are we talking about here?
So these are diagrams of visualization. So how you visualize data. And so you're taking a comparison, kind of a classic example you can see on the internet is like you have a duck playing like a key phone, and then you have a badger, and then in the middle is a platypus playing like a key thing. Like
guitar.
Yeah, so it's guitar, right? So it's like like a badger is playing a guitar, and then the other one is playing a piano, and in the middle you have a platypus playing a keytar. So
right.
the thing is, you have two
I'm
circles.
really confused now.
Oh, now you're confused. All right, well.
I mean, I know what a Venn diagram is, but
right.
I've never seen these platypuses.
You've never seen this one? Okay, well, okay. So basically,
usually they're just two two circles that overlap to some extent, and they try to show like how similar or different something is. But I actually wanted to ask you a question about your research, Richard. If we're
sure.
Okay,
so I'm gonna send the link for the platypus keytar for everybody here, so they can see it. So you just look up platypus Venn diagram, and you'll see the figure I'm talking.
That's amazing.
Yeah, that's the first time I've ever seen that. Oh, that's funny.
If you look, just say literally Google platypus Venn diagram, you'll see what I'm talking about.
We'll add this in the link in the description as well. We'll add this in the description for the podcast.
Yeah. So go ahead and ask that question, Sttephanie.
Well, so you said you're working on the next virus.
I'm working on the next pandemic.
That's what I meant. Yeah. Well,
yeah.
Similar. So, I mean, can you go more into that? Like, what exactly are you doing? Are you preparing like a vaccine so it's ready, or like what do you mean by like you're gonna? I'm assuming you're gonna solve the problem or faster. Sorry, I'm not asking this right.
Well, my kind of main thought is the next pandemic will not be viral.
Okay.
The next pandemic will be multi-drug resistant bacteria.
Mm-hmm.
Think of a time which we all in the room, or outside the room, obviously, when we didn't need. There's been multiple times in my life where I've gotten a bacterial infection in my life, and everything from when I was a child all the way up until I was an adult. And if there wasn't an antibiotic there, I would have died. Right? And so I got scarlet fever one time, and if I hadn't had that antibiotic, I'd have been not here to have that conversation. So we are going towards a place to where you will get a cut on your leg, you'll get a strep throat, you will get a sexually transmitted bacterial disease, and there will be nothing that we can do for you. And so there is forms of gonorrhea now that are multi-drug resistant. There is also that's in humans. The other one that I also fight is a bacterial disease that affects plants. It's called fire blight, and it basically will collapse the pear industry in the United States. There's also fungal diseases in plants. There's a classic one called take-all wheat, and it could become multifungal resistant, and you'd have a whole field of wheat and no wheat in it. And so, and also looking at multi-drug resistance in chickens and, all these types of things like that. And so I'm a virologist, right? So why is a virologist, how could a virologist be going after a bacterial or fungal disease? Well, I'm using viruses to destroy them. So it's an old dog doing a new trick. And so this thing is actually Luria, a very famous phage biologist, bacteriophage. These are viruses that kill bacteria. They don't infect, they don't hurt us. You have more phage in your mouth that protects you and getting in from food than there are stars in the Milky Way galaxy. You have more viruses in your gut than there are stars in the Andromeda galaxy, and there's more viruses than there on the planet, 10 to the 31, than there are stars in the observable universe. So they have this tremendous potential and tremendous genetic repertoire, tremendous library. And viruses probably are the reason we all exist. 20% of all the biomass in the ocean is lysed by viruses every day, right? And so there's also viruses that protect us from disease. There is a phage that prevents lethal doses of alcohol in our body and non-alcoholic fatty liver disease potentially. So nature has developed an Excalibur, a weapon to defeat these multi-drug resistant bacteria. But WHO by 2050, 10 million people will die every year from multi-drug resistance. We've seen an increase of over 30 to 60,000 people every year will die from C. diff, which is an antibiotic-induced disease. Again, gonorrhea, multi-drug resistant. I mean, it's only a matter of time. And think of even plague hasn't gone away. Like Yersinia pestis, it's still around. The only difference between us now and the time of the Black Death was that we have public health measures and we have cleaner water supplies and stuff like that. But, who knows, right? And so 700,000 people die every year from multi-drug resistant bacteria. So we're getting close to where we're losing that battle against bacteria and fungi. And so we can use viruses that don't harm us. They're bacterial viruses, bacteriophage. And we can help people. There's this brilliant book called The Perfect Predator. It talks about this woman and her husband got a cut on his leg, and when they were visiting Cairo, and he got Acinetobacter, was also called Gulf War syndrome. It's a gram-negative beta gamma proteid bacteria, and he almost died. But they found a phage, bacteriovirus, in a river, and it saved his life. And not only using them for therapies in humans, but plants, animals, and these viruses are part of molecular biology. Sequencing, all of that came from phage biology. All the promoters are viral. So we have an opportunity to manipulate these viruses synthetically to make everything from batteries to materials to therapies to new types of food even. So tremendous potential. So then you're asking, then you'll ask, what's the next viral pandemic? What would be the next virus to be a pandemic virus? In that one, if with all the will of my body and being, will likely come from the paramyxoviruses. And I will do, in my group and others around me, now that we've seen this, we'll do everything in our power to prevent that. Because coronaviruses, we said back in 2007, there was a paper that was published that said that this is a time bomb. And we waited too long. We cannot wait on the paramyxoviral family. And so you're like, what is that? So this is the family of measles. Measles, and that family, if you look at you'll hear these things like R0. What is an R0? What is that? So that's the replication unit of the virus. So that means if I am infected, we have COVID, SARS-CoV-2, I infect 3 people. It's an R0 of 3, right? The measles family has anywhere from 10 to 18 people you infect, right? And they have much higher mortality rates. The Nipah virus, there was in Kerala, India, there was an outbreak of Nipah virus in Kerala, India, and it had an 80% mortality rate. The movie Contagion is actually based on a paramyxovirus. Yeah.
Do you know where this virus would come from? Like, would it be from wildlife, from factory farms?
There we go. Wildlife once again comes from bats.
Yeah, bats are the
bats. 25% of all mammals on the planet are bats.
Mm-hmm. Yeah.
Oh, really?
Yeah. So.
Yeah, most mammals are bats and rodents.
Yeah.
Well, for both of you, I think the question I was thinking was, when we're talking about climate change as well, that's going to affect the wildlife. Let's say from the equator up, so maybe on the north coast of Africa, you're going to start getting the migration of animals up to the south coast of Italy, and so on and so forth, as the climate gets worse. So is that then going to, is that an added spanner in the works then, where we look
Absolutely.
at this mass migration of bats and things as they come further and further north?
Absolutely. The other kind of, absolutely, but absolutely. But I want to miss one little bit, but I want to go back just 10 seconds. So what are we working on? So we're working on to prevent, so the goal is to have the next pandemics, multi-drug resistance, or a virus out of the paramyxoviral pandemic they will never happen. That's the goal. Is that most people will be blissfully unaware that they were on the edge of an abyss. If my work is successful. So how do we fight them? We fight them by modifying these viruses to go after these pandemic these. We can even modify phage to block and inhibit other viruses. So we can modify these, and then we modify them as small proteins and make new antivirals, new antibiotics, and new antifungals. They're based on synthetic biology, and so that's the goal of my group. So
I have a question about. Sorry, go ahead.
So hold that one quick. I want to finish what he was saying, and then we'll come back. Okay. So yes, climate change will impact wildlife migration. That's a dead ringer. Another thing that I'm also kind of worried about, there is, actually I think it's outside of Scotland. I was watching something I was reading something about anthrax. Mm-hmm. So there's this one island, and anthrax was used as a bioweapon, and there was the one island that was used as a test point, and no one was isolated the government kind of came in and finally fixed it. But there's thought that one of these, and there was a paper that was published that said, okay, well, they found these giant viruses that don't infect people in permafrost, mm-hmm. right? And that they were still active 30,000 years later. With all of these megafauna that have been frozen, right, for a long, long time, something like anthrax not a viral pathogen, but something like anthrax could be awakened. Mm-hmm. With the thawing of permafrost, like that, and an outbreak could happen. And so we need, again, we can't wait on a vaccine. Vaccines are amazing. I'm 100%, 1,000% vaccine. I'd have vaccine tattooed across my chest if I could. But
you can.
I know I can, but I won't. Vaccines, I believe we should be and I've said this before, my other one with these guys is that vaccines are the knockout punch, the uppercuts to eradication.
Mm-hmm.
They should not be the first line of defense. I think we should be in the business of eradication of disease. We've done this with rinderpest, which is basically cow measles, and we've done this with smallpox. None of us probably in the room have had smallpox, and that's thanks to global efforts to eradicate it. And that's from the people, because it was only in people. But if it's in something like a bat or a fox or a mink or whatever, we need to vaccinate and treat wildlife and have surveillance of wildlife. And we need to be thinking about when this if the megafauna, because even if we started getting carbon deficient and carbon neutral today, we're on a trajectory where a lot of these ice caps will melt. Mm-hmm. And so we need to be thinking about if they do melt, what is our threat of something like anthrax or Eosinus pestis? 1918 flu, the H1N1 flu, was resurrected from frozen Inuits dead Inuits in Alaska. Mm. And that's how they reconstructed the virus. And so there might be viruses and bacterial pathogens that are frozen in those megafauna that we have not seen in 20,000 years and would have no immune response to it, right? And so we need to be investing broadly, both in climate change, scientific education, and surveillance of such events. So then we'll go back to your question. Sorry, Sttephanie. Hopefully you remember.
No, it's okay. This is super cool stuff. My question was, you were talking about the antibiotics. So I know, a lot of it's contributed from the use of antibiotics, like from doctors giving them unnecessarily or from them happening in animal feed. Do you think that reducing the use of them would help at all, or do you think we're past that threshold where it's all out there?
So yes, I don't think they should be used a lot in people that have viral disease, but I don't think the overuse is necessarily in people. I think a lot of the overuse is in agriculture.
Yeah.
And I think that Europe has gotten rid of some of the agricultural antibiotics and asked that exact question. And the answer was the antibiotic residues are still there 10 years later. So even if we stop today, they're still there. And we have basically vaccinated, if you will, these bacteria to be more deadly. Right? And so even if we stopped all of it tomorrow, it'll still be there. However, viruses and making cocktails around multiple viruses have less of a chance of resistance. And so so this is a fundamental property, and this was a property that was discussed between Luria and Fleming back in the 1930s. Luria was phage biologist, yep, this is not going to work in the long term. Fleming is yes, it's going to save a lot of lives. So syphilis syphilis is going to fix syphilis from World War I and World War II, and it did. Syphilis was was one of the main things that helped stop syphilis was penicillin. But within 2 years of releasing penicillin, you have antibiotic resistant bacteria. Mm-hmm. So Luria was right. Even 2 years after this was going, but there's even therapies now. C. diff, sorry, it's not C. diff, CF, cystic fibrosis, is a disease. It's a polymicrobial disease caused by a single point mutation in the human genome to where they make more mucin. They get infected with Pseudomonas from the air and other microbes. It's polymicrobial infection, but Pseudomonas is one of the main biofilm formers in the lung. Phage therapy can be used in the combination of antibiotics to basically make the Pseudomonas susceptible to the antibiotics again. So most of the things with Pseudomonas is they pump the antibiotic out. Antibiotic comes in and it pumps it out. So the virus uses a pump to actually enter that cell. And so what the bacteria does is say, well, I got to get rid of this virus, so I'm going to just get rid of that pump and evolve away from this virus. Now they're susceptible to the antibiotic again. So you can do these kind of evolutionary combo therapies between antibiotics and viruses to basically eradicate some of these pathogens.
Yeah.
I'm
I think
speechless.
yeah, it's exactly because I'm thinking, right, I thought we had 10 problems, but it's it's 50. Even things like things like water quality and things. I mean, this is a huge problem, and it's getting worse and worse. I mean, take that into combination with antimicrobial resistance and all of these things. It's a cocktail for absolute disaster, isn't it?
You guys should explain cocktail though, because people won't know what a cocktail is.
So what I mean by a cocktail in this case is, so classically, this is kind of a stolen word from HIV therapy. So you have HART, or highly active retroviral therapy, and so they call them drug cocktails. So meaning more than one drug. That's all that means. And so and you have a phage cocktail. You have more than one virus, more than one phage. And again, people are like, well, I don't want a virus in my body. And I was like, well, too bad. They're already there in massive abundance. And actually you drink billions of them every time you have a glass of water, and they fall from the sky on you and you don't even know it. And they're your first line of defense. 70% of our immune system is functioning due to the training of the microbiome, and our microbiome is the first, 70% of our immune system, and our immune system trains from that microbiome, and so that's why, these kids that, you have this thing called the hygiene hypothesis, where kids that were kept inside and didn't get exposed to kind of dirt and
I was just going to ask you about that.
Yeah, and so, they have all kinds of odd autoimmune problems. And so now we live in this really odd time. And so Marty Blaser, you can read some of his work. It's really great. And so it's this hygiene dilemma, right? So right now we have a deadly virus that has killed. It's infected 9% of the U.S. population, and has killed out of those 9% that have infected 28 million people. 2% have died. The thing we haven't talked about is all the people that have this idea of recovered, which is absolutely silly, because recovered can be anything from a double lung transplant to basically minor symptoms. And so there will be a lot of people from this pandemic that will have permanent disability, and of the bereavement of the pandemic will be another thing. But going back to your question, Sttephanie, do I use more alcohol on my hands than I ever thought I would in my entire life over this last year? Absolutely. I never would have thought my hands would drink more alcohol than my mouth, but that's where we are at. And yes, but do we run the risk of hurting the good microbes on our hands? And that's a possibility. And limiting our exposure, but we have to right now. We have to limit our exposure to prevent the virus, but then in the future, how do we have this perfect balance between hygiene and not hurting our innate microbiome? And so a lot of the ways that I try to live, and I tell folks as studying the microbiome, is eating a lot of fermented foods. Sauerkrauts, yogurts, they're better than that pill that you take from probiotics. A jar of yogurt has way more bacteria and bacteria than those pills that you take. So, kombucha, fermented foods, I think, are a way to where we have to keep our hands clean right now, and we should be doing that anyway. But then how do we not hurt our own microbiome? We eat fermented foods. The other thing that Marty Blaser has put together is this idea of like you have a seed bank for seeds. He wants a seed bank for microbes of the human body. That way we don't lose them over time. and we find that they are therapeutic, we can put them back in people. And so that's where we have to strike a really fine balance right now between hygiene and safety because of this virus. But I think that while we're dealing with this pandemic, we can eat fermented foods, and that will help maintain a robust and also prebiotic foods. So like lots of complex fibers and stuff like this, and fruits and vegetables will help maintain pre and probiotics in the form of microbes and prebiotics in the form of fruits and veggies. So.
I just read in the New York Times that the transmission of the virus through surfaces is very, very small, and they suggested that we don't need to hyper clean anymore. Do you agree with that?
You know, I need more data personally, so I do wash, and mainly not for the virus. I wash for other bacteria and stuff that are on, fruits and veggies.
Yeah.
And so I do tend to wash. I do tend to clean my stuff. It seems the data is that it is low from transmission from, like, the word is fomite, right? Not, but I still am cleaning stuff because I don't know if someone, obviously some people are wearing masks, but if someone sneezes on that food product and then I go and grab it, even if I clean my hands, I just personally am not going to risk it.
Yeah, I always wash too. I've never used hand sanitizer in my life. Even like in rural field work, I would rarely use it. But here, really the only thing I do is go to the gym, which I know a lot of people think is like the most risky. But our gym is so large, and there's so much space in between everyone. Everyone has to wear a mask. So I just sanitize after when I'm leaving the gym, and then I wash my hands all when I get back at my house.
I wish my gym compatriots were like you, because most of them do not wear masks. I did have to tell a woman the other day to put it over her nose, and the conversation ended with a F off.
Oh, really?
To me, yes.
Yeah.
Because she said she couldn't put it over her nose because she has breathing problems. And I said, well, you shouldn't be at the gym if you can't put it over your nose. And also, if you have breathing problems, aren't you worried about getting COVID?
Yeah, I don't understand that one either at all. Like,
she did.
I saw one time a fellow at Costco with an oxygen tank. And in that case, yes, he can't wear a mask because he have to oxygen tank. But I was also saddened that if he had to go, because that was his only way to go. That was the only way for him to get his groceries, is that he had to go. And that hurt my soul a little bit.
Yeah.
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 I mean, it's funny for me. I mean, I've always hated things like touching, like the poles in public transport or coins and stuff like that. And again, those are two things I've not had to do at all really for the past year. Everything's paid cashless, by card, and I'm not using public transport. But I have been into the department a few times, which is an old 60s building, this kind of brutalist architecture type thing. And after this had all started, I hadn't realised how many double doors I have to open between, walking in the front door and getting to my office, going from my office up to the staff common room to get a coffee, and I realised I was constantly having to open doors, and it's because, it's something you would never ever have thought of before in my entire life, where how many doors I have to open in a building, But now I do, because every time I touch it, I'm going, oh my God, I have to put on the sanitising gel and all this stuff, It's really kind of, to me, it's the small things that have been really upended that have surprised me, if you know what I mean.
Did you have any insider information as to when you think we'll be back to normal?
Nah, that's a good one.
Or we'll be wearing masks forever.
This is a good one.
Because of the next pandemic, too.
So, if you look at the calculations, if you're just doing it on sheer calculations, we're immunizing around 1.5 million people a day. in the United States. Anywhere from, and then if we drop significantly, even we say 750,000 a day, we've officially vaccinated 41 million Americans. Unfortunately, I don't have the European numbers, guys, like the British or the UK numbers. I wish I would have brought it. I should have brought it for you guys today, but I'm sorry. I only have the American numbers. Not that I'm American centric, but I just I don't have the numbers in front of me. So maybe I will. While you guys are talking, I'll answer that question for the British folks that are listening. With that, and then we have around 12% of the population have been vaccinated. I'm predicting by August 1st that we will have at least 70% of the adult population vaccinated. At that point, we are starting to see the effects of herd immunity, which is where a lot of people are vaccinated, and we're starting to see cases drop. We've had 50% of the cases drop in the last 2 weeks, and that's a great sign. There was a American football event known as the Super Bowl, where there was 25,000 people gathered, and this will be a stress test upon and so if we hopefully will not see another uprise of cases. But
I didn't watch. Were they wearing masks and were they separated, or it was just like a normal Super Bowl?
Yes.
Wow. Where was it? What state? Was it in Florida?
This is in Florida. So and I'm just trying to find the correct numbers here. So around 12.2% have been vaccinated with at least one dose, and this is either Moderna or Pfizer, mRNA vaccine. Around 16 million have had 2 doses, and at 2 doses with both vaccines, we're anywhere from 93 to 96%, 94% effective. So with those numbers, I would predict, and maybe this will come back to haunt me, by August 1st that 70% will be vaccinated. Now, everyone is, and they talked to me last time about the B117 variant, which was discovered in England. Viruses have no nationality. It just happened to be discovered in England. And so the B117 variant is protected with both vaccines. So it works on both vaccines. The B1351, which is the South African variant, has a mutation in it that allows for escape of antibodies. They are currently in Moderna and Pfizer are working on a booster to help boost the efficiency. But there was a paper that's recently published, and I'm trying to find that right now, that shows that the early rate of reductions of the vaccine are actually really high, and this is from Israel. And they're actually getting great success with just a single dose. And this was published in the Lancet, actually the 18th, so day before yesterday. So even with a single dose, they're finding, with a recent analysis of a single dose, 80 to 89 to 91% during 15 days after the first dose effective. After you get this first dose of the Pfizer vaccine, 15 days later, you're at 98% protected, which is fantastic.
Yeah.
So, as well, there has been another antiviral that is now heading into phase 2 and 3 clinical, phase 2 first and then 3 clinical trials. And it is an oral antiviral that inhibits SARS-CoV-2 in the lungs, and it appears to be very, very effective. So, and I will try to name the name of this thing, and it's been shown in mice. And then I think we just have to wait for the clinicals to get back to us. But we have a weapon against this virus. And what's interesting is this orally broad spectrum antiviral also goes after Hep C and other viruses. Remdesivir works well. It's very expensive because it's not orally available. It's an intravenous drug. It also was found to work in Ebola. So as we continue to find antivirals, eventually we'll be taking antivirals. It won't be a big deal. Hopefully, we'll still make vaccines. My worry is that we have the antivirals. We won't, the folks who say, let's throw no money in the vaccine because people are getting sick with it. And I want to say, no, there's absolutely value in the vaccine because then we can eradicate it. And so even though it's not killing everybody every day, I don't want people's death to be the only reason we make vaccines. I want us to make antivirals. I want to have a litany of antivirals, and then we want to, figure out what that virus is, or bacteria or fungi, and then build a vaccine to eradicate it. That's where our mindset should be for infectious disease. So this never happens again.
Yeah, definitely.
I want this to be the last. So the antiviral is called EIDD 1931, and it's beta hydroxycyadine. And the prodrug is actually was just published in Nature today. Francis Collins, the director of the NIH, retweeted the Nature paper today. So there is a great potential of new broad spectrum antivirals. I will also say that the Nature paper was out of Ralph Barrett's lab here in North Carolina, Chapel Hill. So hurrah for North Carolina.
All right, guys, I think we're reaching a sort of natural end there. Is there anything
it was nice to finish that on a positive note.
What's that, sorry?
No. Oh, sorry. It's good to finish on a positive note. I was worried that it was going to be all doom and gloom about, the virus and stuff like that. But to finish on a positive note, that we're everything's looking good.
Yeah. So, our vaccine is effective. Numbers are dropping. Deaths are still up there, but they're dropping. We have potentially a new antiviral that will work not only on SARS-CoV-2, but work on other coronaviruses and may even other viruses. It may even open a whole new class of antivirals for us. The mRNA vaccine technology, it's been around for 40 years, but it is a technology that may also be used for a variety of different things, everything from cancer to other autoimmune diseases. So, as we, try to reach the moon against the Russians, the United States, right? What came out of the space race? The device we're talking on right now, the phones that we use, the TVs, all that stuff came out of the space race. And so out of this race to defeat this virus and this disease, we will leave the world better off. And it is absolutely a tragedy that all these people have perished. And not to take anything away from that, but millions in the future will be saved by these vaccines and these new things that we've discovered.
So as Ross says, it was great to end positively because it started off on climate change is going to ruin us. We're becoming resistant to, or bacteria is becoming resistant to antibiotics, so they're going to ruin us. And now but we have vaccines, so yay science.
We're doing well. I just want to
I guess the thought is that...
Sorry.
Go ahead.
Go ahead, Richard, it's fine. Oh.
So my thing is to, again, invest broadly in STEM and education. And we're the good guys. We're here to help. We're not your enemy. We're not, part of some thing to hurt you. We're here to help.
We care about you.
And we care about you.
Exactly.
We care about you. We do this because we do it because we love it, and we do it to prevent loss, most of us. And don't fall asleep on climate change, and we can defeat it. We have the technology. We have the ability to defeat it. We need the will to defeat it. And we had the will to defeat this virus. We have the will to be the first, potentially the only, biological species that reaches the stars. And we can't let ignorance and greed be in the way of that. And we have to leave it better than we found.
Perfect. Perfect ending. I was going to ask if anybody wanted to sort of end the podcast there, but I think that's a perfect way to end right there.
I think so.
Awesome. So,
pleasure meeting you both.
Oh, definitely, man.
It was nice to meet you as well. It's been nice talking to North Carolina. From Fife to North Carolina.
Yes. This has been cool as fuck. Let's just put it out there. This has been awesome.
Yeah, we didn't think this
Thank
was,
you. This is fun.
we
It's
didn't
a lot of
think
fun.
this
I'd
was
love
going
to
to
do
work.
it again.
We were actually very worried that no one at all was going to turn up after we badly interviewed everybody, and we're surprised that a few people did turn up. And for us, it's been absolutely brilliant. We barely said anything. We got a bit confused about what figures were, and it turns out we just misunderstood what was being talked about. But it's been absolutely brilliant. Thanks very much for coming, and hopefully we've helped science a tiny wee bit.
Yeah, a tiny bit. I hope so. I think, I hope we have.
Yeah.
Gotta run, but thank you. Thank you so much.
Thank you both. Pleasure meeting both of you, everybody.
You too.
Man.
Right.
Take care.
Bye.
Right, thanks, lads.
Thanks,
Bye.
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 Bye-bye.
Cheers.